Namaskar everyone, my name is Garvit Sharma and I am a PhD student at International Center for Genetic Engineering and Biotechnology ICGEB. I am working as a PhD student under Dr. Naseem Gaur in Yeast Biofuel Group. At Bharat Innovates 2026, we are going to demonstrate our technology of novel dry yeast formulation for enhanced ethanol production from molasses and grain. India aims for 20% ethanol blending target currently, and the major ethanol production comes from maize, syrup and grain-based feedstocks. The active dry yeast plays a major role in ethanol production from these feedstocks. Currently in India, the majority of the active dry yeast comes from the foreign countries such as China, US and Japan. And talking about the indigenous strains, they suffer from very low ethanol yield and at elevated temperatures. So therefore, at ICGEB we have developed a indigenous thermotolerant and inhibitor tolerant yeast. Now our aim is to convert this yeast into the active dry yeast formulation and to sell to the Indian distilleries. Okay, so during the bioethanol production, distilleries faces several challenges such as contamination, such as low sugar yield and at elevated temperatures. So therefore, the active dry yeast poses a very feasible solution for the bioethanol production from these molasses and grain-based feedstocks. And in India, currently the major distilleries are shifting from molasses to grain-based. So in future, the active dry yeast demand will going to be increased. So therefore, the production of active dry yeast in India is should be the primarily focus for the bioethanol production. At ICGEB, we have developed a Saccharomyces cerevisiae NGY10, which at elevated temperature and high inhibitor stress, and our strain has surpasses all the commercial available strains right now in India. So in our lab, we are producing the active dry yeast using the molasses-based feedstock. We are growing the yeast culture into the molasses medium. We are having the capacity of 5 liter shake flask, and we are growing this yeast culture for almost 3 days. Later we centrifuge it and dry it to produce the active dry yeast. So this is the active dry yeast we had manufactured in our ICGB laboratory, and right now our aim is to scale up this process to a pilot production plant, and soon in 1 or 2 years we are going to plan to sell this active dry yeast to the Indian distilleries. So in our lab we have also worked on the 2G bioethanol production. We have completely optimized the pre-treatment process to treat the sugarcane bagasse or other agricultural waste residues. Here it is shown that we are checking that whether the active dry yeast can able to consume the fermentable sugars released from the agricultural waste. Below, we have engineered the Saccharomyces cerevisiae to ferment the C5 sugar available in the agricultural waste residues into the bioethanol.